Oh, we are almost through with this, and I know it's kind of been long, and sometimes my explanations may not be very clear. So please let me know where you do have problems. So when we talk about proteins, there's actually a whole lot of different proteins. One of the reasons is because proteins monomers, there's 20 amino acids, and these monomers then can be combined in any variety of ways and numbers. So we liken this to the alphabet, right? With 26 letters, we recombine those 26 letters into many, many words. So proteins are the words, right? And this is great because proteins are not going to be generally used for energy. Yeah, they have energy within them. They have calories, clearly, when you consume them. Potential energy, right? But proteins, we've already got our energy covered, right, with carbohydrates and lipids, and then lipids give us some structure and some signaling in the form of hormones. So now proteins are going to do all the other things in our body that we need. And so this means, again, we put them into structures. They're used for our immune system, and all the enzymes we talk about are ultimately proteins. They are transporters, okay? So they are gates, they are channels, the receptors, all proteins, right? So proteins, organic in nature, carbons and hydrogens, oxygens, like the carbohydrates and the lipids had, but now we add nitrogen. A protein will always have nitrogen because the amino acids all have nitrogen, right? And sulfur is found many times in the proteins because there's a few of the amino acids, cysteine and methionine, which contain sulfur, and they are found commonly in proteins as one of the letters, right, in the words. So again, amino acid, in the name, we see this amine terminus, and then this carboxylic acid, or carboxyl, end. All right, so it's a carboxylic acid, and these actually do function as both acids and bases. These can buffer either one because of these ability to accept hydrogens and to release hydrogens. 9 of the amino acids are considered essential. This varies from different sources, but usually that has to do with whether or not there are some differences between children and adult. But as adults, we say 20 amino acids, 9 of which are essential, which means you have to get them from your diet because your body can't synthesize that amino acid. So here, same as we had with the monosaccharides and the fatty acids, we saw that when we put together the monomers, this time again the monomers and amino acid, we put them together, it's by dehydration synthesis. So a water is removed, and we do use this term peptide bond between them, and this is a dipeptide, but really it's just showing that, again, in these anabolic or synthesis reactions, it's really occurring in similar mechanisms. All right? So proteins are put together from these individual amino acids, and they just have these 11 here as a chain of amino acids, and this chain is considered the primary structure. It's the first level, right? So, and this is what your ribosomes do with the assistance of messenger RNA and ribosomal RNA and transport RNA, or transfer RNA, right? These all represent the peptide bonds. Now, these are different amino acids, and so they have different reactive groups in them. I mean, I didn't really point that out much because this isn't a biochemistry class, but each amino acid varies in this group that's found here, from as simple as something like glycine, which is a hydrogen, to something very complicated like tryptophan, which is a double-ringed structure, tyrosine, and so forth. Right? So here, depending on what amino acid is next to which one, we get differences in the secondary structure. The secondary structures have a few motifs. The most common are called the alpha helix and the beta sheets, right, that are shown here. And again, this is determined by what that reactive group was here. And let me just clarify that that reactive group is often, they fall into categories. Get ready for this, right? They're polar, right? Non-polar, right? Acids. Okay, so if you've got some polar and non-polar next to each other, they tend to repel and they form a different structure than if you've got polar and polar, right? Which polar likes polar and non-polar likes non-polar, and polar doesn't like non-polar, right? So we talk about these secondary. So the proteins basically begin to fold, right? and they will fold more. So collectively, these alpha coils and these beta sheets are occurring in these long chains, right? Because proteins are generally 100 amino acids or more, up to thousands of amino acids. So you can have these different motifs in the same basic protein, and so what that sums up to is a third, right, tertiary structure, which is shown here. And then in some of these proteins we get quaternary, a fourth level, and that's if you have multiple polypeptide, right, many amino acid chains. So something like hemoglobin is a good example. It's got 4, right, polypeptide chains that are put together. This is the iron component. The heme unit has iron ions in them, and or atoms in them, and this is a pretty complicated large protein. So all proteins start off as a primary structure. They are then folded, generally in the endoplasmic reticulum, and we'll talk more about those as we get into the cells, into a secondary structures, and continues to be folded into a tertiary structure. So all proteins would have a tertiary structure, and then larger proteins have quaternary structure. All right, so here again, some of what I just said. So this is showing you hydrogen bonds, which is what really these dotted dashed lines are, hydrogen bonds, and this occurs again because you've got this nitrogen and oxygen, which are have high electronegativity, and so they pull the electrons closer to them, and there's lots of hydrogens in these amino acids, and that creates this, again, hydrogen bonding. The tertiary structure comes about from hydrogen bonding and covalent bonds, so a little bit more complex, right? And I say here again, not all proteins have these chains. It's just the larger proteins. Okay, proteins are the controllers, as we said. They do most of the functions because we have energy taken care of. We have all the energy we need to do our reactions. Now we just need we need some molecule to actually do these functions, right? So I mentioned here that they control, and that's the job of enzymes, right? They're catalase, and they control rates of reactions. So we want to introduce this vocabulary, denaturation. And you're going to do this in the lab too. You're going to denature an enzyme, and the enzyme actually has a name, catalase, so don't confuse that with catalyst. Catalyst is a job, catalase is a name, right? And so the enzyme, being a protein, has these levels that we talked about here. And so it has a very unique shape. And we've already figured out that structure reflects function. And because of this unique shape, it then interacts with these other molecules or reactants. And because there's a relationship between a specific relationship between the reactants and the enzyme, we now call the reactants substrates. And as they bind, a change occurs. And this change is mediated by the enzyme, right? And we said, it's because enzymes lower the activation energy for the reaction, and you don't have to remember that, but that's what happens here. And so the enzyme makes or assists in this reaction happening. Again, the reaction would probably happen on its own. Many reactions are spontaneous, but it would take a time, and you don't have time in your body and in staying alive and staying in homeostatic balance. So, and again, this just reminds you, catalase, what I've already said here. So a substrate is a term we use when we're talking about enzyme-controlled reactions, and this substrate is specific to the enzyme. So if this was a different chemical, we'd have to use a different enzyme, right? And this is a very, very specific relationship, and we talk about it as a lock to key or lock, whatever. It's really more of an induced fit, but it's not here or there, because again, I'm really not going to test you on these concepts. I do expect you to know what enzymes are and what substrates are. All right? And so I say refer you to page 77 in the book, and note all the things that proteins do. They do pretty much everything but provide energy. They are found in all living organisms. They are fundamental to life. And I say, what happens to the protein in your diets after you swallow? Well, your digestive system breaks them down to amino acids, and then your body, they are moved into your blood in absorption. They move through your body. The cells pick them up, and the cells rearrange them into anything they want to. Your cells are always doing protein synthesis and all kinds of different protein synthesis. Okay, the last group of organic compounds that are important to us are nucleic acids. And so nucleic acids, the monomers of these are called nucleotides. Okay, the big molecule is a nucleic acid. So go figure, again, organic compounds, so carbon, hydrogen, oxygen, nitrogen, like the proteins do in the amino acids, but not sulfur, phosphorus. This is the only one we're talking about that really always uses phosphorus. Phospholipids did as well, but a nucleic acid, and specifically nucleotides, always have a phosphorus in them. And so what happens is, again, here's that phosphate group that we've kind of zoomed in. This is the base, or nitrogenous base, so named because it has nitrogen in it, right? Attached to, yes, a pentose, a sugar, either deoxyribose or ribose, which is attached to a phosphate. Okay, so this unit is a nucleotide. So this phosphate group and this pentose sugar are pretty much, they're the same. I mean, it's not pretty much, they are the same from nucleotide to nucleotide, right? What's different is this, right? Now, there's not a lot of difference here. As you notice, I've got 5 choices here. So it's not like the 20 amino acids. It's only 5 choices. And it turns out in DNA or RNA, there's 4 of these. So whether you're DNA or RNA, you have cytosine, right? You have adenine, and you have guanine. But if you're DNA, you use thymine as your nucleotide, thymine. And if you're RNA, you use uracil. Now, we always make our lives easy and abbreviate these with single letters, right? And so when we talk about nucleic acid sequence, or DNA sequence, or what you might call the gene or the genome, we're basically looking at a string of A, T, C's, and G's. And when we talk about an RNA molecule, like messenger RNA, transfer RNA, or ribosomal RNA, we're looking at a chain of, right, C, U, A, and G. Okay? So RNA and DNA are the only examples of nucleic acids we're going to talk about, right? Well, there's 200,000 different proteins and counting. There's only RNA and DNA here, right? So they do differ in their sugars. This is a structural aspect. And they differ in this base. And then the third difference is that RNA is generally found as a single chain. Remember, we talked about the quaternary structure of proteins being multiple chains. And DNA is a double chain, right? So DNA, RNA, and ATP, right? So we add the ATP here, not because it's a nucleic acid, but it's fundamentally it's got some similarities, right? So we have adenine, that base, right, which is, again, a purine, so double-ringed. Pyrimidines are single-ringed, right? We'll come back to that later, but that is something to keep those words in your head. A ribose, right, in this ATP, and then 3 phosphates as opposed to 1. This is really important because these are very high energy bonds. All this negative charge pushed together is like cocking a spring. It's really loaded, right, and very unstable, right? So 3 phosphates, so triphosphate. All right, now, just to give you, again, something to think about, there are approximately 22,000 genes that we've identified as human genes, divided into 46 chromosomes in every nucleus of cells, all right? And the exception is that you have 23 chromosomes in your sex cells, and you also don't have nuclei in your red blood cells. These carry the chemical codes for making proteins. That's why they're so valuable to us. And these chemical codes are transferred from a DNA molecule, nucleic acid, to an RNA molecule, and then a ribosome basically decodes them or translates them, right? So ATP, I said here again, we've already talked about, uses a nucleotide with an exception, right? It's got these extra phosphate groups. This is used for energy, and we make this in every cell because every cell requires energy. All right, so we'll come back and play a Kahoot to see if you're getting this in our class. All right, we're done with chapter 2. Time to move on. Hope this is kind of making sense. Please come with any questions or send me questions. Thank you.